Welcome to our video lecture on topic 9.4 plant reproduction. Our essential understanding for today, reproduction in flowering plants is influenced by the biotic and abiotic environments. Our objectives, we're going to review some genetics, we're going to identify vegetative and reproductive structures, we're going to talk about how the flowering of angiosperms is controlled by gene expression, which is why we're doing that genetics review. We're going to talk about the functions of different parts of flowers. We'll outline pollination, which is an example of symbiosis, so we'll go ahead and review that too. We're going to talk about the functions of different pieces and parts of seeds, talk about how those seeds get dispersed, and then talk about how those seeds germinate. First up here is some of that genetics review vocabulary that I just mentioned. I am not going to go through all of these words, but pause the video, take a look at this, and then restart the video. All of the definitions are on the next slide. Here are all of those definitions. Go ahead and again pause the video if there are any that you need to jot down, review. Otherwise, here we go. So flowering in our angiosperms involves a change in the gene expression of the shoot apex. Remember that the shoot apex is up at the top of the plant. We're going to change what genes are being activated, what genes are being inactivated. That's going to lead from a switch from growing vegetative structures, roots and stems and leaves, instead to reproductive structures, those flowers. Vegetative structures have indeterminate growth. They can just keep growing and growing kind of indefinitely, as opposed to the reproductive structures, which have a very pre-planned determinate size and shape, and they just grow to that and then they're done. So how do the plants know when to flip from indeterminate growth, stems and leaves and whatnot, and instead start to grow those determinate pieces, the flowers? It's all about influences of that abiotic and biotic environment. I'm going to say lots of words on the slide, and then all these words are going to pop up on the next two slides, but printed on the slides instead of my just saying them. So don't panic too much about following everything right now. I'm going to kind of come back and say it all again. You'll get to read it all again. So again, here we go. Abiotic environment, we're going to talk about sunshine the most here. So we've got these short day plants and these long day plants. You can probably guess that short day plants bloom when they have short days, and long day plants bloom when they have long days. But a bad piece of the naming of these plants is that it's not about the short day plant, because notice that if I give my short day plant a short day and a long night, but I interrupt that night with a little bit of sunshine, we end up with no flowers. And then in my long day plant, if I give it a short day, it really wants a long day, but then that long night I interrupt with a little bit of light, so really it's getting two short nights, I get those flowers. So short day plant, we should better name long night plant. And our long day plant is better named short night plant. How does this lack of light influence genes? It's all about these proteins called phytochromes. So phyto for light, chrome for color. Phytochromes come in lots of different forms. We're going to look at PR, phytochrome red. Phytochrome red is our inactive version. It is the more stable version. It is what we get at nighttime when it's not been activated by the sunshine. In the presence of light, this guy changes shape a little bit and turns into phytochrome far red, far red, far red. This guy's a little bit less stable. It's the more active version, and the more light we have, the more PFR we get. But when we take the light away, it's going to turn back into the PR, the phytochrome red, more stable. Less stable with the light, more stable without the light. More during the light, PFR, less during the light, PR. So in our short day plant, more PFR is going to turn off the flowers. So more light, more PFR is going to shut down the flowers. As opposed to in our long day plants, the more PFR, the more far red we have because there's more sunshine, we're going to actually activate those genes that turn on the flowers. And as promised, here are those words again, but in words on the screen instead of just my talking at you and looking at pictures. So gene expression is going to control flowering. We have lots of cell signaling involved. The stimulus for the signal is usually the length of night. We have phytochrome red being converted into phytochrome far red during the day, going back to phytochrome red at night. The more night we have, the less phytochrome far red we have. The more day we have, the more phytochrome far red we have. That phytochrome far red is going to be that phytochrome pigment that influences this gene, the flowering time gene. That flowering time gene, when it's turned on, will be transcribed. We'll make some messenger RNA, which will then be translated on the ribosomes, and we get some flowering time protein. That protein is then going to bind to activate some genes. Those genes will transform. The apical meristem will stop growing stems and leaves, and instead we'll start growing those flowering meristems at reproductive determinate tissue. Because that activation or inactivation of those genes being transcribed, being translated, depends on the amount of light, we call this photoperiodism. Period being the length of time, photo being the light or the lack of light. So photoperiodism, how much light, not light, are we getting? And that's going to influence that gene expression. It's all dependent upon that phytochrome plant pigment. We talk about phytochrome red, which is going to absorb red light. That's why we call it phytochrome red, right? And it's going to be converted into phytochrome far red. In the darkness, without the energy from that red light, phytochrome is going to go back to the more stable version, phytochrome red. So light turns red into far red. In the darkness, far red goes back to red. Phytochrome red is never, ever, ever active. It is stable. It doesn't do anything. It does not bind to any receptors. Phytochrome far red is the guy that's always active, but here's where it gets weird. In our long day plants, phytochrome far red is a promoter and says, hey, flowers, let's grow. But in our short day plants, more phytochrome far red is going to turn off or inhibit the transcription of those flowering time genes. And so too much far red, too much sunshine shuts down those flowers in our short day plants. Lots of far red, more sunshine, more far red, turns on the genes. We do get the flowers in our long day. Too much far red, too much sunshine, too much far red is going to shut down transcription, translation of the flowering time genes. So we don't get flowers in our short day plants. Good. And here are all those ideas one more time. Phytochrome red is that pigment in plants that in the presence of red light, and there's red light in sunshine, or all our grow lamps, that phytochrome red is converted into phytochrome far red. Phytochrome far red is the active version that will either turn on or turn off those flowering time genes, depending on if you are a long day plant or a short day plant. Long day plants get promoted or activated by the phytochrome far red. In our short day plants, it is inhibiting. That phytochrome far red inhibits flowers. If I have a short day plant and I interrupt its long night with a little bit of light, no flowers. If I have a long day plant and I give it just a short day, but I interrupt its night and give it a couple short nights, voila, we end up with the flowers. So Bio Ninja again with the win. Sid the lad is how we remember phytochrome far red, phytochrome red. Short day plants, the phytochrome far red inhibits flowering. That's our Sid. And then lad in long day plants, phytochrome far red is going to activate that flowering. So let's say that we have successfully activated our flowering time gene and we have flowers. Flowers are the reproductive structures of plants. Of course, there's a stem. The stem connects the plant to the flower. Lots of vascular tissue in here. We need xylem and phloem to carry water, minerals, organic materials to the flowers so that it can do all of its super cool stuff. Sometimes we'll add here at the base of the flower, the top of the stem, a peduncle, which is just an enlarged portion of the stem. Sometimes the nectary is also here, the nectary just where nectar is formed to attract those pollinators. Petal's job also to attract some pollinators. The stamen is the male portion of the flower. Anther at the top is where we produce some pollen. Sperm is in the pollen. The filament is going to hold that anther up to make it easier for the pollinators or wind or however that pollen is being spread to access that pollen. The pistil, which is sometimes also called the carpel, is the female portion of the flower. At the top of it, we have the stigma. The stigma is sticky. So try to remember sticky stigma. It is sticky so that pollen will literally stick to it. The style holds the stigma up so that it will run into that pollen, make it easier for the stigma to grab onto that pollen. And at the base of the pistil, we have the ovary. In the ovary, we have the ovule, and in the ovule, we have some eggs. What's pretty cool, the ovary eventually will grow into the fruit of these angiosperms. Pollination is literally just the spreading of pollen from the anther of one stamen to the stigma of a pistil. Pollination can come in a couple different kinds. We have cross-pollination. This is generally preferred by plants, where we are taking pollen from one anther and taking it to the stigma of a different plant so that we can increase genetic variation in that species. Some plants can also carry out self-fertilization, where pollen from the anther will stay in that same flower and go to the stigma of that same flower. We're not increasing genetic variation, but we still get plant babies, so that's a win. Pollination is often carried out by pollinators like this glorious bee. Notice all the pollen covering his body. He's sticking his head in there to get to the nectar. The pollen is just going to stick to him. He's going to fly to a different flower to get more nectar. That pollen is going to fall off his body. Some of it at least will fall off his body onto the stigma of that next plant, and that pollen gets spread. Pollination is an example of symbiosis, and so I figured we might as well do some symbiosis review. Remember that sym is together and bio is life. So we're talking about organisms that are living together that depend on each other for whatever it is that they need. Predation is an example of symbiosis. This is a positive negative. One thing gets a snack, the other thing dies because it is the snack. And our example of predation is not the crocodile and the plover, but instead is the ladybug devouring this aphid. So ladybugs are serious predators. They will kill and eat those aphids. Aphids are like mosquitoes on plants. They will stick their little noses into the phloem of plants, suck out the nutrients. Gardeners do not love aphids, but gardeners do love ladybugs because the ladybugs will eat the aphids right off your plants. That aphid is dying. It is going to get eaten. It is not surviving. Parasitism is similar in that it is a positive negative. Something gets a snack, but the other thing loses some nutrients but doesn't die completely. An example here is the mosquito feeding off of the host human. Aphids feeding off of the plant is also an example of parasitism. Mutualism, this is where it is mutually beneficial. Both organisms get a benefit. This actually is the crocodile and the plover. I know it looks from the picture like that bird is about to get gobbled up by the crocodile, but instead what's happening is the crocodile just holds his mouth open and the bird literally picks out meat from between the crocodile's teeth. The crocodile has healthier teeth, which allows it to eat more and live longer and make more babies. That's a bonus. The bird gets a snack. The bird doesn't have to hunt and kill anything. It can just literally take the scraps out of the crocodile's mouth. So this is mutualism. Both are getting a benefit. Pollination is also mutualism. The bee gets that nectar and the plant gets pollinated. The last kind of symbiosis that we're going to look at is commensalism. This is where one gets a benefit and the other one is neither helped nor harmed. It's just there. It's neutral. And the example is here with the cattle and the egrets. So these egrets are not actually eating any ticks off the backs of the cows of the cattle, but instead what happens is they just hang out by the feet of the cows. And as the cows walk through the grass, bugs are disturbed by that walking, and the bugs will jump away from the hooves of the cattle. Jumping bugs are easier for the birds to see, and so the birds will snack on the grasshoppers and whatnot that are jumping away from the feet of the cattle. The cattle are not helped at all by the birds, but the birds definitely have an easier time finding food because they hang out by the feet of the cattle. Again, pollination is that symbiotic relationship. It is mutualism because both organisms benefit. Pollination is simply that transfer of pollen from an anther to a stigma. It can be self-pollination if the anther and the stigma are in the same flower. It can be cross-pollination if they're in different flowers. Generally, this is preferred because it means genetic variation within the species, which is good for survival of the species. But again, I mean, some seeds are better than no seeds, so self-pollination is a really good backup plan. Fertilization occurs after pollination. So pollination, we get that pollen grain on the stigma of a pistil. That pollen grain is going to grow a pollen tube down the style, that's that long neck of the pistil, through the ovary and into the ovules. There are sperm cells in the pollen grain. Those guys are going to go down that pollen tube and into the ovules where the eggs are. The eggs will be fertilized. What's kind of cool about the pollen grains, they actually have 3 sperm in them. One's going to fertilize the egg, and then other ones are going to fertilize endosperm, which is going to be the food that is kept in the seed to help that baby plant grow. And once fertilization occurs in those ovules, they will develop into seeds. Here we have a couple different kinds of seeds, lots of variation. These are just a few examples. Seed coat on the outside of many seeds is there for protection. We also have radicle is the baby root, and plumule is the baby shoot. Radicle, plumule, sometimes here we're going to call it the hypocotyl instead. Here we've got radicle, we've got plumule, so that's the baby plant. Also inside the seed we've got some either cotyledons or endosperm, or sometimes we have both. So the job of the endosperm cotyledons is nutrition. So here's where we're storing lots of food, lots of starch for the baby plant to have lots of energy to make lots of ATP to do all the growing that it needs to do. And we're going to zoom in here at one particular seed because sometimes IB will ask us to draw and label a seed. So here's a really nice one to draw. Things to add: the testa, also called the seed coat. Its job is protection. The cotyledon here, or it could be endosperm in different kinds of seeds. Its job is nutrition. So lots and lots of starch, amylose, amylopectin, stored here for the baby plant to grow. The epicotyl or the plumule is the baby shoot. The radicle is the baby root. The micropyle here is a little hole where the pollen tube had grown into the ovule before the seed was formed. What's pretty cool is that there is still a little bit of a hole left there, and that's how water will get into the seed so that germination can occur. Once those seeds are developed, the plants are going to try to spread them out or disperse them. The goal is for the baby plants to not have to compete with the parent plants for light and water and nutrients. So spreading the seeds out allows for the prevention of that competition. Lots of different modes of seed dispersal. It could be wind, if you've ever played with the dandelion fuzz or the maple helicopters. Super, super fun. These guys get pushed around by wind. Animals, these burrs can stick to the fur of animals and the animals will walk around and then the burrs can fall off. Some things will deposit their seeds into water and then the water will carry those seeds away, deposit them someplace else. Some things will just rupture. So when we humans walk past violets or touch-me-nots, they'll just like explode and then move around that way. And then of course, us humans have developed lots of crop plants and we will disperse the seeds ourselves. The last little piece that we're going to look at here is germination. So you've got a seed. How do you get a baby plant out of that seed? There are lots of different factors that will affect germination. Water, of course, is super important. Oxygen, these are needed for ATP production. We also have some important temperatures and pHs that are involved. Some seeds will have specialized conditions where they will only germinate after a fire, or after it's been frozen, or some guys have to be eaten and pass through the gut of an animal and then get pooped out before they will start to germinate. The different processes involved in germination, we're going to always have to start with some water. That water is going to wash away one kind of hormone on the outside of seeds called ABA, or abscisic acid. It is an inhibitor of seed development. We do not want seeds to start growing before the conditions are right. So ABA will keep that seed dormant until it is time. Water comes along and will wash away that ABA so that the seed is no longer held dormant. The water will also stimulate activation of a gene that is going to lead to the production of this other hormone, gibberellin. The gibberellin is then going to activate the production of the protein amylase. That amylase in the seed will then start to break the amylose, if I could spell, amylose down into lots of glucose. That glucose is used by the mitochondria in the seed to make lots and lots of ATP. That ATP then will allow for all the metabolic processes to happen. Generally, the root, the radical baby root, will pop out first, and then we end up with our cotyledons. Those are those seed leaves. Tiny little baby leaves from the plumule or the epicotyl will pop out next, and voila, we have a baby plant. And with that exciting accomplishment, we have completed our video lecture on reproduction in flowering plants, which is influenced by biotic and abiotic environments, especially that sunshine. So we talked about a little bit of genetics, we talked about vegetative, the indeterminate growth of plants, reproductive structures are determinate growth. We talked about the control of flowering by gene expression. We have phytochrome red and phytochrome far red. We have that flowering time gene. In our short day plants or long night plants, that PFR is going to inhibit flowering. In our long day plants or short night plants, phytochrome far red is going to promote that flowering time gene. And then we end up with some beautiful flower structures. We talked about the petals and the anthers and the stamens and the sticky stigmas, the styles, the ovaries, the ovules. Pollination is that transfer of pollen from anther to stigma. It is an example of symbiosis. We talked about different functions of seed structures. Make sure that you can draw a seed. We talked about different ways that plants can spread their seeds out so that the baby plants are not competing with the parents for resources. And then, of course, how do those seeds sprout is germination. Great work today.